Welcome. In this session, firstly, I'll discuss the detailed structure of the plasmid PBR322, and then later we'll see vectors for cloning genes in plants and animals, and thereafter we'll also see about competent host. So let's begin this session with the detailed structure of this PBR 322. So I hope in the last session I already told you what the meaning of this PBR 322. So this is a plasmid, right? So let's see the structure of this plasmid, right? So this is the structure of PBR 322, and whatever here I have mentioned, so they are all the restriction endonucleases. Uh, so which will be going to act on that particular region. So in the tetracycline gene region, so they are two recognition sites. So already in the last session I told you what we mean by the recognition site. So the locations where the restriction endonuclease, okay, cuts, that is what we call the recognition site. Of course, first it identifies the palindromic sequence that is specific to this BAM H1 or SAL1 or PVU2 and so, right? And of course, these are different restriction endonucleases that we have obtained from different bacteria. So like for example, this CLA1, it is obtained from a bacterium called Caryophanon latum. So is the bacterium from which we obtain this CLA1. And the HIND3, this we have obtained from Haemophilus influenza, is the bacterium from which we have obtained this restriction endonuclease. Okay, then the SAL1, so it is Streptomyces albus. Okay, then the BAM H1, so this is from the Bacillus amyloliquefaciens, is the bacterium from which we obtain that BAM H1 restriction endonuclease. Already we know that echo R1. Then PVU1, this is obtained from Proteus vulgaris, is the bacterium from which we obtain this. Then the PST1, so this is from Providentia stuarti. Okay, right? Of course, this PVU1 and PVU2 both are from this Proteus vulgaris. So this is the first restriction endonuclease that we obtained from this PVU1, and this is the second one from that Proteus vulgaris bacteria, right? So these are the different recognition sites that we see in case of this PBR 322. So I told you that the ampicillin and tetracycline are two selectable markers with which we can able to identify the recombinant and non-recombinant. And now let's see these restriction endonucleases, what type of palindromic sequences. We know that each restriction endonuclease will identify a particular palindromic sequence. So what that palindromic sequence is, it's going to identify. Okay, in case of this SAL1, so it will be identifying, okay, GTCGAC. So this is the palindromic sequence that it identifies, and it's going to cut between the G and T. So I just mentioned here something like this. So which means there will be cut between, okay, I'm sorry. So G and T out of these palindromic sequences. So I have mentioned only on one strand of this sequence. Actually, there are two. I hope you already know the palindromic sequences. If you come across, come from this point, that is from the right to left, also it will be GT, okay, CG, and AC. So the cut will be between G and T, right? And coming to this PVU2, it will cut between, right, CAG, it will cut between CAG, right, okay, and then of course this is the palindromic sequence. It's going to identify this CAG, then CTG is the palindromic sequence, and it's going to cut between G and C. So CAG and CTG, right? So in this case, this is going to form a blunt ends. So it forms blunt ends. So in this case, the SAL1 is not going to form the blunt ends. It's going to form the cohesive ends or sticky ends. So here in this case, the SAL1 is forming, right, sticky ends. So it forms sticky ends. In this case, PVU2, you can see on both strands, it is going to cut on the same point. So for that reason, we will be getting blunt ends. Then coming to PST1. So PST1 will be identifying a sequence called CTG and CAG. So the PST will identify CTG and CAG, and this is going to cut between the A and G. So that's, I'll mention CTG and CAG. Okay, and coming to this PVU1, of course in this case also it's going to form the sticky end. And coming to this PVU1, so it's going to identify, right, CG, AT, and then CG, right? So of course the opposite will be also in the same. And now in this case, this PVU1, it's going to cut between, okay, T and C. That's, I'll mention CG, A, T, and C. So between the T and C, it's going to cut. So that's what in this case also it's going to form a sticky end. So here also sticky end. So this one forms, you know what you say, the blunt ends. Already we know that echo R1. So I already mentioned that GATTC, you know it. Okay, I'll not mention that. Then coming to this BAM H1. So this BAM H1 will be GGATCC. It's going to identify a sequence called GGATCC, and it's going to cut between G and G. Okay, again forming sticky ends. If you are using this BAM H1, it's going to cut, recognize this palindromic sequence and cuts between G and G. Then coming to HIND3. So in case of HIND3, it will be, okay, the palindromic sequence AA and then GCTT. So this is the palindromic sequence identified by the HIND3 enzyme, and it's going to cut between A and A, right? And the last one, the CLA1, okay? So this CLA1 is going to identify the sequence AT, CG, and AT. So it's going to identify AT, CG, AT, right? And the cut will be between the T and C. So that AT, C, G, 80. So that will be, the cut that takes place between, so after recognizing this palindromic sequence by the CLA1, it will cut between T and C. Right, so that's the structure of this PBR 322, and I told you that these ampicillin resistant and tetracycline resistant genes will act as the selectable markers and will help in identifying the recombinants and non-recombinants. So how is that, we will see in detail. So let's see here, if I'm going to insert the alien DNA, of course the alien DNA, foreign DNA or gene of interest, whatever, one and the same. So here I have taken, okay, the alien DNA inserted into this BAM H1 site of tetracycline resistant gene. So here I have inserted, so there is an insert, the gene of interest. So now as I inserted this alien DNA or gene of interest, what happened is the sequences of this tetracycline resistant gene have changed. Okay, say suppose the gene of interest has a sequence of GATC something. Okay, of course it will be not only that much short, it will have many more bases. I'm just taking a small example. Now say suppose the tetracycline resistant gene has something. Okay, so this is the sequence here. Okay, and of course we need to put in between the BAM H1 site. So the BAM H1 site will be, say it will be GG80CC. So that's the sequence. So the BAM H1 will identify the palindromic sequence, the GG80CC here. So when I apply this BAM H1 restriction endonuclease, we know that the BAM H1 is going to cut between G and G. So then it becomes AT, GCC, AT, then G, right? And then this one gets separated. Okay, so into this I'm going to put this gene of interest here. So that will be an insert here. So that's what I'll mention with a different color, so we can identify. So GATCC, GAT, and then the next continues the GATCC, AGTATA. So this is what it becomes. And we know that these nitrogen bases, whatever, so in the genetic code they will be read, okay, 3 together. So this 3 will form or code for amino acid, this 3 and this 3 will form okay, another amino acid. But what happened here, okay, the sequence after this is going to change. So as a result, the tetracycline gene becomes inactivated. The tetracycline gene, okay, gets inactivated, okay, due to insertion, and of course that is what we can call it a insertional inactivation, right? So it becomes inactivated tetracycline. If you do it in the ampicillin, then the ampicillin will become inactivated here. So at present I have done it, okay, linking the alien DNA into the BAM H1 recognition site of tetracycline gene, and this gene became inactivated. But ampicillin resistant gene is still working. So this is working, but this will not work. Now what I'm going to do is we need to put this plasmids. So that is, say this is the PBR 322 plasmid. we need to put that in the bacterium. Okay, we need to transform the bacterial cells and then take the media, so that contains this antibiotic. So this is a medium containing ampicillin. Okay, so medium containing ampicillin, so that is antibiotic. And I'm going to take another medium containing tetracycline, now into this I'm going to put the bacteria that has, okay, the plasmid with the gene of interest, so that is the rDNA it has, so you can see here. And now in this case the bacteria that doesn't have, okay, the gene of interest. So this, okay, is a what I say, the E. coli bacterium. So in this case we'll say E. coli bacterium that has, the plasmid with the gene of interest, so that is rDNA it is having. So I say this is actually transformant, is a transformant. Okay, and of course this is also a transformant, but this is having non-recombinant DNA, so it's having non-recombinant DNA. So that will be not having the gene of interest. So here, if it is not having that, then the tetracycline will be working. So here in rDNA, the one which has taken the gene of interest, in which the tetracycline is not functional, but in the non-recombinant DNA, so the non-recombinant DNA, these two selectable markers, ampicillin resistant and tetracycline resistant, so both are working, so both will work. Okay, so hence if I put this, the E. coli bacterium having recombinant non-recombinant DNA in a medium containing tetracycline, it will live, it won't die, because in that tetracycline resistant gene is working or it's functional, so hence it produces the proteins that can counteract the action of tetracycline, and this bacterium will be live. And now coming to the this one, the E. coli that contains the gene of interest, in which this tetracycline is not working. In this case, this will live in, so transform it with rDNA, so transform it with rDNA, and this is the transformant, okay, right, with the non recombinant DNA, the transformant with non recombinant DNA. Now this will live in ampicillin antibiotic, because in this antibiotic is functional. But if I put this one in the tetracycline, right, it will not be able to live, this E bacterium will not be able to survive, because in this the tetracycline gene is not working, and because of that it will not able to counteract the action of tetracycline, and it will die. The transformant with non-recombinant DNA, it will live in both the media. Okay, so that's, so this transformant with non-recombinant DNA lives on both media, whereas this transformant with rDNA can live only in ampicillin containing medium. live only in amphisilin containing medium. Okay, this will not be able to, okay, live in this that is what the non-recombinant I mean lives on, okay, or I'll say lives in both the medium. So it's able to survive in the medium containing tetracycline and also in the medium containing ampicillin, whereas this one, okay, the transformant with rDNA will not be able to survive in a medium containing tetracycline, in a medium containing tetracycline. Okay, so with that we can identify so which have taken the gene of interest and which is not. But the point is that maintaining this simultaneous plating of this with ampicillin and tetracycline antibiotics, and this procedure actually in fact a cumbersome procedure, means it's a very difficult to identify the recombinant and non-recombinants. So for that reason, so scientists started finding out a new way, and they have come up with another solution. So what is that? Instead of this antibiotic resistant genes using as selectable markers, so they started using, okay, enzymes as selectable markers. So let's see. So they have taken a plasmid in which there is a sequence, right, which will be coding for an enzyme called beta galactosidase. So say this is the region of that plasmid that codes for beta galactosidase. Okay, so it's an enzyme. Now this enzyme will actually help as a selectable marker, and of course will help in identifying the recombinant and non-recombinant. And when I'm going to insert that gene of interest into this sequence that codes for the beta galactosidase, so I have inserted this sequence. So this is the alien DNA that I have inserted, and similarly what I have mentioned earlier, that when I put alien DNA into the tetracycline resistant gene, it's going to become non-functional. In this case also same happens. If I insert the alien DNA into the beta galactosidase gene, then this beta galactosidase gene will become inactivated. So this portion, whatever that the gene coding for the beta galactosidase, becomes inactivated, and that's what we call it insertional inactivation. That's already I have mentioned earlier, insertional. So due to insertion of alien DNA, the gene becomes inactivated. Okay, now what I'm going to do, I will be taking a medium that consists of a chromogenic substrate. So chromogenic substrate. So the meaning here, what is this chromogenic substrate? It is going to produce color upon reaction with the enzyme. So that is beta galactosidase. Now this is a recombinant. Now in this case, okay, recombinant, the beta galactosidase is non-functional. Okay, so in recombinant, beta gal is inactivated or non-functional. And if you see with another one, okay, this is another plasmid. In this, right, the beta gal is functional. Beta gal is functional. I told you in the previous sessions that when we take these plasmids, okay, in a test tube, and then we will also take this alien DNA, okay, so alien DNAs, and then we will be using restriction endonucleases to cut open the plasmids and then later DNA ligase, so that the plasmids will get ligated with this, but the point is that all the plasmids that cut open will not be taking that gene of interest. So some will take and some will not take it. So that's what where we have come across with the recombinant and non-recombinant. Now, if I'm placing, okay, the bacterial colony, so here this is the transformant in which it has the beta galactosidase inactivated, and this is the bacterium in which it has, or the transformant that has, the DNA, the plasmid, which is not recombinant. So it's having non-recombinant. So I say the transformant with the non-recombinant DNA. So which means in that beta gal is active. So in this case, the beta gal is active, and in this case the transformant with the rDNA, in this case the beta gal is inactive, that the beta gal is inactive. So as a result there will be no beta gal enzyme. So if no beta gal enzyme, then there will be no reaction with the chromogenic substrate. No reaction with chromogenic substrate. So when there is no reaction with the chromogenic substrate, it won't produce any color. So no color, right? So the transformants with rDNA will be colorless. Now coming to the transformant with non-recombinant DNA, in that the beta gal is active. In this case, the beta gal enzyme is present, and that will react with the chromogenic substrate, will react with chromogenic substrate. So when it reacts with the beta galactosidase enzyme, when reacts with the chromogenic substrate, it's going to produce blue color. It's going to produce blue color. Then we can with the color only we can identify. That is what we did not see in earlier method, where we have used antibiotic resistant genes as selectable markers. So in this case, this, okay, beta galactosidase will be the selectable markers. Okay, so that's a selectable marker. Fine. So I hope this is clear. So we will be seeing on this chromogenic substrate, one colony of bacteria which will be colorless, and another colony of bacteria which will be colored, that is blue color. And when you see the blue color, we understand that those are the bacteria who are having a non-recombinant DNA. So means which did not take the gene of interest. And in this case we see the bacteria where the beta galactosidase gene became inactivated. So hence there is no enzyme generated or expressed, and because of that, no reaction with the chromogenic substrate, and there is no color. Okay, so the one colony with no color indicates that is having rDNA, and the other one with color, that is blue color, are non-rDNA, right? This is how we have changed from selectable markers like antibiotic resistance to the enzymes like beta galactosidase, right? I hope that part is clear. Next, we'll see about the vectors for cloning genes in plants and animals. So how to deliver the genes into plant cells and animal cells. Okay, of course, in case of these bacteria, we have used those plasmids. So we linked that gene of interest to the plasmid, and okay, allowed the bacteria to take up that plasmid. So when the bacteria takes up that plasmid, it becomes transformed. Right, now how we are going to transform the plant cells and animal cells, and what kind of vectors we are going to use, okay, to deliver a gene of interest into the plant cells or into animal cells. So that's what we see, vectors for cloning. Vectors for cloning genes in plants and animals. So vectors for cloning genes in plants and animals. Okay, so here we have come to know, right, there is a bacterium called Agrobacterium tumefaciens. So a bacterium named, okay, Agrobacterium tumefaciens. So this bacterium, when it infects the plant cells, it causes a disease called crown gall, or I can say it will cause tumors. So it causes tumors in plants, right? So I can say it is, the one that causes these uh, crown gall. So I say these tumors in plants as crown gall in plants. So it's a disease. So this Agrobacterium tumefaciens, when infects the plants, it will cause tumors in the plants. So those tumors, of course, we commonly call it the crown gall in plants. Now, how this Agrobacterium is able to cause that tumors? So this Agrobacterium tumefaciens has a plasmid. So it has a plasmid, okay, and this plasmid has genes, right? So that actually make the cell to become tumor. Means the cell will be going on dividing. See, tumor is a condition when a particular cell in which there are certain kind of genes which will regulate the cell cycle, means how many times the cell need to divide. If anything happens to those genes, or those genes are suppressed, the cell will not be able to control how many times it need to replicate. So it will go on dividing and forming a number of cells. That is what the tumor formation, how it takes place, right? Now this plasmid, that is the Agrobacterium tumefaciens plasmid, that is specifically called Ti plasmid. So this is Ti plasmid. So where this Ti stands for tumor inducing. So it induces tumor. So tumor inducing plasmid. And this tumor inducing plasmid has a region. So it has a region that's called the tDNA. So this is called the tDNA. So where the T in this case stands for the transfer. So this is the DNA that it is going to inject into the DNA of the plant cells. So thereby, so this tDNA starts making enzymes that will alter the cell cycle of the plant, and the plant cells may start going on dividing and result in the formation of tumor. Okay, so it means that the agrobacterium knows how to introduce its genes into the genome of the plant cells. So what we have done is we took this Ti plasmid from this Agrobacterium tumefaciens and removed this tDNA. So we remove this tDNA because this tDNA will be causing that disease crown gall. So we removed it and we altered this plasmid and we made this plasmid so that it will carry, right, my interest genes, means whatever the genes what I like I can, okay, link to this plasmid and make this plasmid to deliver that genes into the plant cells. Okay, I hope that is clear, right? So just mention, so this Ti plasmid is altered and made it non-pathogenic, non-pathogenic. It is no more, because when we remove this segment tDNA from this Ti plasmid, it will be not able to cause the crown gall or the tumors in the plant cells no more. So instead, okay, I'm going to link the genes of my own interest and make this Ti plasmid to deliver that genes into the host genome. Okay, so that's what, how I'm trying to transform the plant cells. So that is what the cloning vector in case of this in case of plants is this Ti plasmid. So this Ti plasmid is a cloning vector for plants. It's a cloning vector for plants. Then what we are doing with the animal cells, how to transform the animal cells. So for these animal cells, okay, we are using something called retroviruses. So in case of animals, we use retroviruses, right? So these retroviruses are they are disease causing, and the retroviruses, the viruses that have, okay, RT enzyme. So they have RT enzyme, which means it's a reverse transcriptase. It has reverse transcriptase enzyme. So what this reverse transcriptase enzyme is going to do, this reverse transcriptase enzyme is going to make DNA from RNA. We know the transcription process, okay, where RNA will be formed from DNA, but this reverse transcriptase is going to make DNA from RNA. So that process we call reverse transcription, and the enzyme involved in the reverse transcription is reverse transcriptase. So those viruses who have this retro you know who have this reverse transcriptase enzyme, they are called retroviruses. I hope in human health and diseases you might have come across the HIV virus. So HIV virus is a retrovirus that contains reverse transcriptase enzyme. So most of the viruses, those viruses who have RNA as the genetic material, so they will be having this reverse transcriptase enzyme. Right, now what we have done, so we have taken these retroviruses, okay, and we altered their sequences. So we have altered them, or we disarmed retroviruses. So we disarmed the retroviruses, and after disarming the retroviruses, okay, so we are able to link the gene of interest to these retroviruses, and because viruses have that ability to deliver the genes into the host DNA or host genome, okay, so for that reason I will be using these viruses and make them by removing. So we know all we have, how to cut, how to paste, the molecular scissors what we have with us, the restriction endonucleases, and the molecular stitches like DNA ligase. So you can cut, paste, however you want. You can alter the DNAs of any organism. So we have taken those retroviruses, disarmed those retroviruses, and these disarmed retroviruses are used to deliver gene of interest into animal cells. So for transforming the animal cells, we are using the retroviruses. Whereas in case of plant cells, we are using the TI plasmid of Agrobacterium tumefaciens.